Good afternoon, everyone. Welcome to Gresham College. I'm Chris Dye, the Professor of Physics, and this is my third series of Gresham lectures in which I'm going to discuss the relationship between disease and health, the environment, genetics and culture, which is just about everything. So I'm feeling pretty unconstrained in this series of 6 lectures. The first of them is going to be about race, genes, and disease, as you can see from the title up there. So why that and why now? Well, race, of course, is a subject that ignites debate in any context, and health is no exception. And we don't want to just have debates about race. We actually want to raise questions, and we want to find answers to those questions. At the same time, we're living in the throes of a genetic revolution which will proceed through the 21st century, and that genetic revolution promises to put the discussion about race on an entirely different footing. It will contribute towards a much better public understanding of the relationship between genetics, race and disease, and that of course is what Gresham lectures are all about. So to kick off with, I rather like this photograph that I came across recently. Clearly, it's a picture of obesity, but what I like about it is it's obesity without the skin color. It's obesity that focuses on some of the aspects of the real biological aspects of obesity, such as the distribution of fat around the human body. In this picture, it's a thermogram, a heat image, in other words. And in this picture, white and yellow are hot, and blue and black are cold, and you can see that where fat is distributed around the body, like the areas behind the arms, the image is cool because heat is kept within the body. And these kinds of images are a good way of focusing the discussion on aspects, the biological aspects of obesity, without racial overtones and racial connotations. We do, however, have to face the racial discussion about obesity and obesity-related diseases such as diabetes. Now, there's a strong relationship between obesity and diabetes. Not all diabetics are fat, and not all fat people have diabetes, of course, but there is a strong association. And the observation has also been made, the provocative observation has also been made, that diabetes rates have different prevalences in different racial groups. These are data for the population of the United States, and you can see from this graph that American Indians and blacks in the United States have substantially higher prevalence rates of diabetes than other groups in the population. So why is that? Is race in this context a valid basis for having a scientific and medical discussion? What explains the observations that you see in this picture? Is it to do with the genetic distribution in these different racial groups, or is it something to do with the environment that they're living in? One of the ideas that's been proposed to explain the current obesity crisis, and that's a reasonable term for it, is the so-called thrifty gene hypothesis. And it starts with the observation that for millions of years throughout human evolution, we have lived through periods where famines were common. And under those circumstances, it's a good idea to have genes that are very good at storing the energy available from food. In other words, genes that are good at storing fat. But it's been a long while, at least in terms of modern history, since famines have been common. In Western Europe, we haven't had famines regularly for 200 or 300 years, but we do still have the genes, of course. And the high-energy food that we're accustomed to consuming today, working with those genes, may help to create obesity. That's one possible explanation for why we have these genes, such genes. If we do, what kinds of genes are they? When that hypothesis was proposed back in the 1960s, we didn't have the genetic tools to be able to investigate it. We now have those tools in order to do that. So we can take a genetic approach to the investigation, but we can also investigate other aspects of the causes of obesity and obesity-related diseases. But what I want to do in this talk is I want to acknowledge that there's a racial component to health, but I want to try to put it in context for you, in genetic context and in cultural context, but particularly in genetic context, which is my main theme. And it's not just, here's another example, it's not just, of course, about susceptibility to different diseases. It's also been found that there's a relationship between the way in which you can successfully or unsuccessfully treat diseases in different racial groups. This was a very controversial paper that appeared in the highly respected New England Journal of Medicine in 2001 about treatment for a heart condition. You'll see there's lots of jargon in the title of this technical paper. The particular heart condition was left ventricular dysfunction, but the essential observation was that this treatment, the so-called ACE inhibitors, worked better in white people than they did in black people. And that, first of all, within the confines of the pages of the New England Journal of Medicine, generated lots of controversy. People lined up on one side of the debate or the other. At the top here, you see one group of authors that were arguing that we should examine health on a racial basis. There are advantages to doing that. Whereas Schwartz at the bottom there is arguing staunchly against that. And you can see the rather pejorative title of his article includes the word profiling, which is already loaded. It's a word like surveillance in current society. Profiling. You don't have to read the rest of the paper. You know what he's going to say just from the title of that article. And not surprisingly, the debate has leaked out of the medical literature into the more popular literature. Witness here Joseph Graves' book he works in the southern United States, called The Emperor's New Clothes, an investigation of the biological theories of race at the turn of the millennium, in which he tries to put race, genes, health and other aspects of race in context. One of his observations that I quote at the top here is genes for drug activity, and in this particular context he's referring to that study in the New England Journal of Medicine. Genes for drug activity don't seem to be localized in socially defined races, an observation which is perfectly correct, of course, as you will come, I hope, to see. So this is what I'm going to do in this talk. I've done number one already, I hope, which is to try to sketch in for you the background to the race, genes, health controversy, and you'll be aware of other aspects of that controversy yourself. The second part, how genes vary among races, is really about the relationship, two aspects of the three-part title, the relationship between genes and races. And then the third and fourth parts are about the relationship between genes and diseases, and in the fifth part I'm going to try to put all three back together again, and in the context of asking the question, what does our genetic understanding about health mean for you and me and all of us today and in the coming years? It's also worth me saying what I'm not going to talk about in this lecture, which is the ethical and moral aspects of the relationship between genes and disease. For one thing, I'm not qualified to do that. I would rather leave that to my colleagues, one of which is Baroness Deach, and she'll be speaking here at 6 o'clock this evening. And if you come to that lecture as well, I think you'll find that there are some points of contact between what she says this evening and what I'm saying in this lecture here. So, part two then. How do genes vary among races? What is the genetic basis for race? Well, when you look through the literature on this subject, the scientific literature on this subject, which is supposedly the most objective kind of literature, you find that there are various studies that emphasize similarity, and there are other studies that tend to emphasize differences. I'll show you a couple of examples where the differences are emphasized in the first instance, and then I'll move to the ones that emphasize similarity. This is one that emphasizes the difference. These studies, incidentally, are not necessarily biased, although one or two of them are. They just have different purposes. This is a study of the genes of people from all over the world, and it's an attempt to classify the relatedness of individuals from different populations by measuring how close they are in genetic terms to each other. And the way you portray that is in terms of this tree. So two people who are on the same branch of the tree are obviously more closely related. People who are on distant branches of the tree are less closely related. What you see at the bottom, you won't read the small print, it doesn't matter. What you see at the bottom, people from sub-Saharan Africa are different from the other groups of people. In fact, they tend to be a little bit more remote than the other groups are. The other groups are a little closer to each other. Now, the precise explanation for that is not entirely clear. That kind of distribution goes with linguistic differences in populations, and it also may be, in genetic terms, a reflection of the fact that people originated in Africa, and as they evolved, they moved out of Africa, and then there was a genetic radiation after they moved out of Africa. That might explain, it might help to explain, why African populations tend to be a bit more distinct on a further out branch than other populations. Here's another study that was a remarkable study that was published only a few weeks ago in Nature, which has used the full power of genetic techniques available now to look at the differences and similarities of populations within Europe. And as you can imagine from this picture, people who are of the same colour, that is, whether they're red, I'm not talking about black and white now, I'm talking about whether they're colour-coded as pink, red, blue, green, yellow, or what have you, tend to be more genetically similar to one another. And as you can see, within Europe, remarkably, people who live within the same countries tend to be rather more genetically similar to one another. That may not be too surprising to you, but what is remarkable is the scale on which this genetic similarity and difference actually happens. What they found by examining 3,000 Europeans, each of whom had their genes examined at 500,000 different genetic sites, that's the power of genetics now, they found that they could take half of those individuals and place them within 300 kilometers of their reported origin. And that is an astonishing degree of genetic evaluation, of fine-tuning of these genetic techniques. So this is a study that shows how genes mirror geographical differences within Europe and points to similarities as well as differences. Here's another genetic study which tends, in my perception, to emphasize the similarities rather than the differences. This is a study of the so-called genetic haplotypes. Now, there are a few words like this I'm going to drop into this lecture. Haplotype is one of them. I hope this will enrich your vocabulary. Haplotype is a word that you should remember. You can drop it into dinner conversations, as in, for example, only the other evening I was up in the bathroom examining my haplotype when all of a sudden, and you can finish the rest yourself. What a haplotype is you'll know that your DNA consists of two strands. Haplo just refers to one of those strands, and a haplotype refers to a cluster of genes, similar genes, on one of those strands. And it's been found by geneticists that cutting up the DNA like that and classifying it into those kinds of genetic haplotypes is a good way to examine genetic variation in populations. In fact, it's such a good way to examine genetic variation that it was the basis of the so-called International Hap Map project, where thousands of researchers around the world are constructing these haplotypes, examining the way in which they vary in populations and how they're related to different aspects of people who live in those populations. But the key result I want to point to here is those three overlapping circles, which show for European, Asian, and African populations that there is a huge degree of genetic overlap between those populations. There are some differences, and once again you can see that the pinkish African circle drops a little farther away from the Asian and European circles, just like the branching diagram I showed you earlier, but by and large, there's a huge amount of similarity between these different populations. Here's another study that was published just a couple of weeks ago as well, which examines genetic differences between different castes and tribes in India. And I like the study enormously. What I am less fond of is the interpretation that the authors have put upon it. You can see on this map the location of the different tribes that were studied in India, 20 or 30 of them. And what they're examining are what they call M and M2 genes. M actually stands for mitochondria in this study, but it might stand for mother as well, because mitochondria are inherited, or the genes in mitochondria are inherited down the maternal line, not the paternal line. So you get these genes from your mother, not from your father. What they have emphasized is that if you look across India at different tribal groups, you can find tremendous similarities between them. So if you look at the yellow flecks, you can see that there are a high proportion of those M genes which cut across linguistic and cultural barriers. And the linguistic barriers between North and South India are enormous. These are totally different kinds of people, apparently, from a cultural perspective today. But genetically, they have these very strong similarities. And that's what the authors of this study emphasized. V.R. Rao, who was one of the authors, is quoted as saying, biologically, there are no castes and tribes in India, only communities. Well, that's true up to a point, but there are, of course, differences between these tribes and castes as well. So we really have to, in these kinds of studies, acknowledge both the similarities and the differences, and not try to shoehorn all the information that we have into one kind of theory or another kind of theory. We have to live with the variation. And one of the nicest and simplest representations of that information comes from Richard Lewontin's book on human diversity. It's a very old book now, but it has the basics just right, despite all the new genetics. And it says that this is how, if you partition all the genetic variation in human populations, so 100% of it, how is that distributed? And the answer is a bit like the overlapping circles that I just showed you. If you take two people within the same ethnic group, and that's the smallest classification, they have about 85% of all the variation you'll see in the population. So they have an enormous amount in common. If you look between ethnic groups, but within broad races, within broad racial groups, that accounts for about another 8% of the variation. And then if you go to the scale above that, and you look between major races, Africans, Asians, Europeans, and so forth, that accounts for the remaining 7% of the variation. So the message that you get from all of this is that we're very genetically similar to people who are in different populations. Now, if genes are the underpinnings of disease, which is what I'm really talking about here, if genes are the determinants of disease, then we'd expect that any genetic defect responsible for a disease is going to be very widely shared across different racial groups. And that's a key point in this lecture. So that was part 2. Let me now move to part 3 and say something about the genetic basis of disease. Now, one of the obvious racial differences in populations is skin color, and some would say that skin color is rather subjective. This became controversial in the current American election campaign when Hillary Clinton's campaigners were accused, presumably with Photoshop, of making Barack Obama look rather darker than he is in certain lights. So there are different representations of Obama depending on what your political perspective is. And they're heavily skewed one way or another. But there are actually more objective ways of evaluating skin color. And this is a map of the distribution of skin color in peoples around the world. It has the obvious distribution that you would expect. Darker purple represents darker skin color, and that's concentrated in the tropical latitudes and away from the temperate regions. And of course, there's a biological reason why skin color varies across these different populations. One of the reasons is that we need to have presented on our skin the right amount of light, not too much, not too little. If we are presented with too little light, we develop conditions, or we did historically, like the one at bottom left here, rickets, which is a vitamin D deficiency, which leads to misshapen bones during the growth of young children. Nowadays, this is easily rectified by diet, but vitamin D is also made with sunlight as well. Of course, if we're exposed to too much light, then you have conditions like the one at the top, various kinds of skin cancer. And so what's happened over millions of years is that we've evolved to have the right level of skin pigmentation in order to prevent being exposed to too little light or to too much light. So this is, on the right-hand side, a cross-sectional diagram through the skin, And Africans, you can see from these black flecks, obviously have more melanosomes, as they're called, more pigmentation. Asians a little bit less, and Europeans a little bit less still. So that is clearly a genetically determined racial association, skin color. But what I want you to notice at the top of this slide is that of the maybe 30,000, 40,000 genes that we have active that make us work on a day-to-day basis, maybe somewhere between 5 and 50 of those, a very tiny, tiny proportion, are associated with skin color. So we have a very striking measure of racial difference between populations. It's the first thing that you would always notice, and yet the genetic basis for that depends on a tiny, tiny fraction of the genes that make us work on a day-to-day basis. So if that kind of thing is determined by very few genes, is that how the relationship between genes and diseases work in general? When we think about genetic diseases, if I asked you about, give me an example of a genetic disease, I won't do that, but I suspect that a number of you would come up with diseases like the ones on this slide here. These are diseases which usually have a simple hereditary mechanism. They're usually due to defects in 1 or 2 genes only, and they're rather rare and they're very severe. So cystic fibrosis is one, about 1 birth in 2,300. Muscular dystrophy is another, about 1 in 3,000. And Huntington's disease, which is represented by the picture at the back here, even rarer than that. The picture at the back is it's actually a beautiful diagram of a rather gruesome process, which is the destruction of the cortex on the surface of the brain, with degeneration going from left to right, where the red color represents mild degeneration of the cortex and the yellow color very severe degeneration of the cortex. And that degeneration, as you probably know, is associated with, at first, rather jerky movements, which are called chorea, Huntington's chorea. And then there are behavioral differences in the late stages of Huntington's disease, which can, of course, limit life expectancy. And limiting life expectancy, this is another key aspect of this lecture, of course, means that you may not pass on the defective genes that you've got to the next generation. And I'm going to say a little bit more in a moment about whether genes persist or whether they disappear from our populations. The earliest evidence, I think this is a fascinating little example, the earliest evidence of a hereditary genetic disorder was found in this fossil obtained by Israeli scientists working in Ethiopia. It's a fossil of a disease. The fossil picture is the inset, but you can't really make head and tail of the actual photograph unless you're an archaeologist. But there's a modern representation of the condition is the color slide. This is called amelogenesis imperfecta. Of course, the medics will come up with a term that make it incomprehensible to the general population. What it means is a fault in the development of your enamel on your teeth. And that means that the teeth, the enamel may be very thin, it may be very brittle, or it may have a pitted structure. But of course, because teeth are rather well preserved, you can look at dental defects in the fossil records. And this is the oldest example of what is known to be an hereditary disorder. It's generally very rare, but for some curious reason, there is a pocket of people in Sweden where the condition is very common, about 1 in 700 people. That observation is quite well known in the dental business. You can chat with your dentist about it next time. But there isn't a good explanation for that. But it's more complex than this. We can identify genetic disorders that are down to 1 or 2 genes, that are clearly purely genetic diseases. So you've got the gene, you get the disease, and you get it in that form, and it's pretty clear. But most genes don't work like that. The problem for analysis, in any context, including the context of health that I'm interested in here, is that what we see, the characteristics of individuals are determined both by their genes and by the way in which those genes interact with the environment. And that makes it rather complex to analyze. Now, you know the expression peas in a pod, which are 2 people that are supposed to look quite similar. It's a rather unfortunate metaphor when you start taking it down to genetics, because for one thing, the whole hereditary basis, how genes are passed on from one generation to the next, was uncovered by a monk called Gregor Mendel, who worked with peas in pods and showed why those peas in pods could be different shapes, and that's the photograph on the left, why they could be smooth and why they could be wrinkly. In other words, why they could be the same or why they could be different. And of course he didn't have any genetic method of analysis except breeding plants, but he worked out essentially the basis of inheritance by his observations on peas. Another way in which you can investigate, a way that's been used for a long time, is to pursue the idea of peas in a pod through analysis of human twins. And the interesting possibility here is that we can compare twins that are identical and twins that are non-identical. Twins that are identical, of course, they come from the same fertilized egg, should have the same characteristics, and any characteristics that they do have in common actually have a genetic basis, because their genes, of course, are completely identical. So if, as in the diagram on the right-hand side, the graph there, if we compare individuals that are identical twins and those that are non-identical twins with respect to the frequency of different kinds of conditions of ill health, and you'll see them ranged along the bottom there. If we take alcoholism, for example, bottom left, you can see that the identical twins, which are black, and the non-identical twins, which are the white bars, are rather similar. And that suggests that alcoholism as a condition doesn't have a strong genetic basis. But if we go to autism, which is the third collection of bars from the right, you can see that there is a very big difference between identical twins and non-identical twins, and that suggests that there's a very strong genetic basis to autism. It's a strongly inherited disorder. And the way in which we classify this in genetics to allow for the gene-environment interactions is to say, let's have an index that we call heritability, and if heritability on a scale from 0 to 1 scores 0, then there's no genetic component to it, it's all environmental. It's about what you're exposed to. If it scores 1, then we're back to the classic genetic diseases. It's almost purely genetic in its control. Now, sometimes pictures are easier to see these things with rather than graphs. At the top there, I've got a comparison, a slide I've borrowed from another source. You'll see at bottom right, non-identical twins at the top, and you can see that they're rather different, those pairs of individuals, whereas the individuals at the bottom are identical twins, and of course you can see that they're very similar in terms of body build. And body build is something I'm going to come back to in a short while. Now, in order to investigate whether genes control anything, diseases included, as I've already said, we have genetic techniques which are now immensely powerful. We don't have to rely on plant breeding. We don't only have to rely on twin studies, but we can screen vast quantities of genetic material and look at the relationship between genetic defects and health defects in individuals. And this study, published in Nature last year, was one of a kind, an absolute technological tour de force involving collaborators from all over the world. What these authors did is the genetic equivalent of carpet bombing. Essentially, you just sequence massive sections of the genomes and you compare diseased individuals and non-diseased individuals, and you look for differences between them. So they compared 14,000 people with illnesses of various kind against 3,000 healthy volunteers. In each individual, they looked at 500,000, 500,000 genetic markers. They obtained in the study 10 billion pieces of genetic information. And what did they come up with? Well, the results are, frankly, rather disappointing. They found 10 new genes associated with 7 common conditions, the conditions that are listed on the left-hand side of the screen here: Crohn's disease, diabetes, coronary artery disease, hypertension, arthritis, and a mental disorder, bipolar disorder. And not only did they find very few genes associated with these conditions, but the ones that they did find had relatively small effects. So a genetic risk of times as you see at the bottom there, 1.2 or 1.5, that's a 20% or 50% additional chance of suffering from this condition. That's a very small level of risk associated with these genes. So it's an extraordinary study biologically, technologically, and in terms of collaboration, but it's not very encouraging with respect to the possibility of manipulating these defective genes. There are other studies that have, however, been more successful, and one of the striking ones published recently was this study of risk of lung cancer in association with copies of a certain cancer gene. Now, as I've already said, you have 2 strands of DNA, and you have a version of each gene on either strand, so you can have 1 copy of the gene or you can have 2 copies of the gene. In this instance, where people have 2 copies of the gene, that's about 10% of the population, and they smoke continuously for long periods of time, these people have an extraordinarily high risk of developing lung cancer, a lifetime chance of about 1 in 4. And that kind of result coming from genetic studies is much more encouraging, with the possibility, for example, of screening. Individuals can screen themselves to see whether they carry 1 or 2 or no copies of this gene, and therefore evaluate their risks of different kinds of diseases given the environment that they live in. This study was played up enormously in the press. In fact, in a surprising way, in some of the scientific press, even the Associated Press ran the title, "If you can't, quote, quit smoking, blame your genes." One of the other aspects of this that really intrigued the popular imagination, in other words, the press, is that the genetic basis for this may be not only physiological. In other words, if you smoke physiologically, you have a stronger chance of doing damage to your genetic material and then developing cancer, but it may also, these genes may also control your behavior. In other words, the process of addiction. And that really captured people's imagination. It's a result you might have read of, but the evidence that these genes control your behavior with respect to addiction is, I would say, not yet watertight. So the question may now be occurring to you that if we have bad genes, that is, genes that have effects on our health, detrimental effects on our health, why do we have those genes? If people die as a result of genetic illnesses, then why don't they simply disappear from the population? I want to give you 1 or 2 insights into why that may be. First of all, we talk about genes running in families, and some of the most celebrated examples of genes running in families are those that run in royal families. If we think about haemophilia, it's commonly known as Queen Victoria's gene, the title of a recent book, or Queen Victoria's disease. In fact, she didn't have the disease, but she was a carrier of the disease. When you think of Queen Victoria, you normally think of something like the picture top left, but in fact, genetically speaking, Queen Victoria was a bit like this. This individual, this woman in the diagram of inheritance at top left there, she was a woman who was a carrier of the haemophilia gene. So she had 1 good copy of the gene and 1 bad copy of the gene. The problem with that is that if she then mates with somebody who has another copy of the gene, or one of her offspring does, and those 2 genes come together in the same individual, then you get a haemophiliac. And her 8th son, I think it was, who was Leopold, Duke of Albany, if I remember rightly, was an individual that carried 2 copies of the gene. He had a fall at the age of 31, and he died of internal hemorrhaging, so he didn't pass on those 2 copies of his gene to anybody else. But because she was the carrier, and because some of her children were also carriers, the gene has continued through the royal family. And Queen Victoria, of course, was grandmother to most of the royalty of Europe. Alexis was her great-grandson. His father was Tsar Nicholas II, and he came of age at around about the time of the Russian Revolution, and there's been much speculation among historians that his haemophilia may have played a part in the downfall of the Russian royal family. Well, this is speculative, there isn't good evidence, it's barroom talk among historians, but it's an interesting story, and you can read the book Queen Victoria's Gene if you want to know more about that. But the interesting biological question here, and what I want to get at in this section, is if haemophilia kills people when they fall over and injure themselves, then why are there haemophilia genes in our populations? Well, in order to answer that, you have to think about when a mutation like that arises, how long can you expect it to last in a population? And there are two aspects of haemophilia that would partly explain, at least, why the gene persists for such a long time. One of them is the carrier status of Queen Victoria. So she had the gene, but she wasn't affected by it. So it's silent in some individuals, and that's a way in which it can be passed on. And the other thing about royal families, of course, is inbreeding. They all breed with each other. And what you want is fresh blood, new genes, people who don't have haemophilia to mate with members of the royal family. And that's a way in which the genes can be diluted out of the population ultimately. But extensive inbreeding in the royal family is, I suspect, one reason why these genes have persisted in those families. Actually, I can find no definitive account of why haemophilia has the frequency it does in the current population. Maybe someone's worked it out, but I don't know who they are. There are other, much more convincing explanations for why we have bad genes in populations, and some of them are to do with tropical diseases like malaria. One of the best-known stories is about sickle cell disease or sickle cell anemia. Individuals who have two copies of this gene form red blood cells like the ones at bottom left here. They're not nicely round, plump red blood cells that you see on scanning electron microscopes, but they sickle. And if you have two copies of this gene, you don't survive very long. But if you have only one copy of the gene, if you're a carrier, like Queen Victoria was for haemophilia, then you don't suffer from sickle cell disease, but you are protected from malaria. And that protection against malaria is one of the reasons why these genes persist in these populations. And it's one of the reasons why sickle cell disease has become racially stereotyped, of course. Because in the United States, for example, where most instances of sickle cell disease today are in black people of West African origin, the gene is associated with that racial type. In fact, if you go back a little bit further, the gene is also common in European populations because malaria used to be common in European populations. So it's common in Greek people, people from Yemen, and other parts of the Middle East. And that then highlights the danger, one of the dangers of racial profiling with respect to diseases that have a genetic basis. Here's another malaria story that's also quite well known. The maps show you the distribution of the two most common types of malaria that people suffer from. The one at the top is called falciparum malaria, which is the most severe form of the disease, most pathogenic form of malaria, and people who suffer from that form of malaria commonly die if not treated. About 1 in 3 people will die if not treated of malaria. And the blue patches on the graph show you the worldwide distribution of falciparum malaria, and they show you in particular that it's very common in Africa. But if you go to the bottom and look at the distribution of the more benign form of malaria called Plasmodium vivax, that's what the parasite is called, you'll see that it's absent from Central and West Africa. And the reason for that, physiologically, is that Central and West Africans lack a receptor on the surface of their blood cells called the Duffy type, the Duffy antigen receptor, or DARK, or just Duffy for short. And because they lack that receptor, malaria parasites can't get in to their blood cells. So that's another racial association. But what we want to know here is not just the racial association, we actually want to know what the physiological explanation for it. But there's a very interesting twist to this story that's come only in the last month or so, and that's about the relationship between Duffy and HIV/AIDS. Duffy, incidentally, in case you're wondering, is not the popular singer of the Welsh race. Pictured at bottom right there, Duffy was the name of the person, a haemophiliac actually, who in the 1950s was first discovered to have this blood type. What's also now known, apparently, or has recently been proposed on the basis of studies of US Army personnel, is that people who lack that Duffy type, they're protected against malaria, but they're more susceptible to HIV infection, and more susceptible by a significant factor which the authors of this study suggest might explain about 10% of HIV infections in Africa. Now, my colleagues at WHO, who are HIV experts, are reluctant to accept this story on the basis of present evidence, but it's an interesting story nonetheless. I've already mentioned cystic fibrosis, and that's another interesting genetic disease, well known to have a genetic cause, but why are there genes in populations? People who have 2 copies of a cystic fibrosis gene die at an early age. They don't pass those genes on. They disappear from the population. Well, the possible answer is that, again, people who carry 1 copy of the gene are protected against other kinds of diseases. And for a long time it was thought that those might be diarrheal diseases like cholera. But an intriguing recent idea is that it's actually tuberculosis. They're both chest infections, of course, and if you have 1 copy of the cystic fibrosis gene, there is evidence that you are somewhat protected against developing tuberculosis. And that would be, since TB was a major cause of death, if you weren't protected against TB, there would be a very high death rate, and that therefore could be a mechanism by which this gene for cystic fibrosis has persisted in the human population. The racial connotations of this are confusing once again. TB was called the White Plague because it was a plague of white populations, but those white populations were also the same populations that suffered from the Black Death. So you've got the White Plague and the Black Death and white populations. It's all very confusing if you rely on the standard terminology. And this is the final example, is one I was published last week actually, and I loved the story so much I couldn't resist it. It has nothing to do with human disease. But for those of you who believe that there can never be pleasure without pain, you'll love this one. This is about a tropical fish called a swordtail, and it turns out that females have ways of recognizing males that they like. And one of the things that females like about male swordtails is the spots on their tails. And males that have spots meet a lot of females, they mate with a lot of females, and they produce a lot of offspring. The snag is that those spots are caused by cancer genes. And those cancer genes, like the individual male in the bottom, after these fish have reproduced, they grow, they cause cancer, the tail eventually rots, the fish can't move anymore, and it dies. But it's already reproduced. It's already passed on its cancer-causing genes to the following generation. So, sex was marvelous, but there's a high price to pay. Let me come to the next section about what we can do about genetic defects in populations, and I want to try to bring race back into the discussion. Genetic defects, the reason we're looking for them, is that it suggests that genetic engineering might be a way to fix it, or genetic manipulation of some kind. So how could we carry out genetic manipulations? Well, there are broadly 3 ways in which we can do it. The traditional way is selective breeding. That's what we've done with farm animals, pets, agricultural plants. We've done it for millennia. Selective breeding. But it's not a very accurate way, it's not a very quick way of getting genes that you want and genes into populations and genes that you don't want out of populations. So the new genetics allows various techniques of genetic manipulation. We've known for a long time that radiation and mutagenic chemicals can manipulate genes, but not very effective. It's usually detrimental. In that list, there are much more promising candidates. Viruses, I'll give you an example of that in a moment. There are transposable elements, so-called jumping genes that move around the genome. We can manipulate those, but not very effectively. Gene targeting, where we can identify a gene, knock it out, and replace it by another one. There are cloning techniques, and so on and so forth. And then right at the bottom, the thing for the future is, once we know what a gene consists of in terms of its DNA, then we can potentially synthesize those chemicals from scratch. We can make genes from scratch. And indeed, there are people who are already beginning to attempt the process of constructing entire organisms from scratch. Simple organisms, of course, but constructing entire organisms from scratch, genes and all. So what I want to do is give you a few examples of where gene therapy has been successful very recently or shows some promise. Now, those of you who are brought up on Thomas the Tank Engine will know about the fat controller. Well, this is not the fat controller, but the fat controller. The fat controller is leptin, which adjusts the so-called adipostat. It's like a thermostat, but it manages fat in your brain. Now, not all obesity is caused by simple genetic defects, but there is one kind of obesity that is, and that's due to leptin deficiency, called congenital leptin deficiency, and there's a simple mutation that's responsible for that. And what you can do to fix that is not fix the gene itself, but the gene produces leptin. So if you provide individuals, young children, who have this condition with leptin, then you have the gene product. And the results are totally miraculous. This child on the left-hand side at age of 3 weighed 42 kilograms. With leptin therapy, at age 7, he only weighed 32 kilograms. It's the same individual. It's an utterly remarkable transformation. The problem is that, and it's a fabulous result for new genetic analysis, the trouble is that this gene is very rare, as you'd expect it to be on the basis of what I've already said. And most genes that are associated with obesity don't work like that. But for the individuals where it does work like that, the possibilities are just fantastic. Now, there are other things that have been tried as well to manipulate the way in which genes act without actually changing the genes themselves. And this is another very recent example on prostate cancer. This is an example where apparently in a preliminary study, diet and exercise intensively with people sticking to a regime was a mechanism by which prostate cancer genes were turned off. So what these researchers did, the study was based in San Francisco, is they took 30 individuals who were suffering from prostate cancer and compared them with those who were not suffering from prostate cancer. They put them on a strict diet, strict exercise regime, and the results are shown in the graph. But the details don't matter so much. What you should take from the graph is that it's a measure of cancer gene activity, if you like. So bars that go up show activity that was enhanced by this process, and bars that go down show gene activity that was reduced. And you can see that in the majority of these individuals, the gene activity was downgraded or downregulated, as they say in the business. So this kind of environmental manipulation of a cancer process shows promise for the introduction of therapy, for managing a process without actually solving the underlying genetic problem. This is a more intriguing example in terms of genetically based disease, because it doesn't affect the gene product like leptin, it actually tackles the gene itself. And this is a process that uses a virus called an adenovirus in this case. You put the gene that you want into the virus, you put the virus in the place you want it in the body, and you hope that the virus is going to introduce that piece of DNA into the faulty cells that possess the defective gene. The condition is one that most of you probably haven't heard of unless you've got a friend or relative who suffers from it, Leber's congenital amaurosis. It's a relatively rare condition, but this kind of gene therapy with a virus as a basis seems to work reasonably well in diseases of the eye. And one of the reasons for that apparently is that you can target genes in eye cells, the retina in this case, more effectively than you can in some areas of the body. Now, this hasn't completely restored the sight of these individuals, but the therapy has certainly improved the sight, the retinal function of these individuals, and that's an aspect of gene therapy that shows some promise. Now, where there are genetic advances of this kind, enterprise will surely follow. You can now have your gene sequenced for about $1,000 in the United States, and there are specialized companies like 23andMe that will sequence your genes or investigate your genes. They will screen your genome, and they'll tell you what diseases you're likely to be at risk of. If you're not sure why it's called 23andMe, you'll see in a minute. Their website's very good, actually. This is the page on breast cancer. It's actually very informative. They don't deal with the most important breast cancer genes, that is most important, the ones that are known to have the strongest effects in providing risk on breast cancer, BRCA1 and 2, which I'll say something about in a moment. They deal with the genes that are more common that have less powerful effects. This is the answer to the question of why 23andMe, if you haven't already got it, from the cover of Matt Ridley's book on genome, the autobiography of a species in 23 chapters. Of course, humans have 23 pairs of chromosomes. The cover is 6x4, which is 24, and that's because they've split the sex chromosomes into two at the bottom right-hand side of the picture. So that's why it's called 23andMe. If any of you want to take up this service, there are a few words of warning to be issued at this point. Google's co-founder Sergey Brin was widely advertised recently to be a carrier of a mutation that causes Parkinson's disease, and he knows this because he paid the $1,000 maybe to have his genes screened by 23andMe. So having discovered that he was a carrier of a gene that might cause Parkinson's disease, first of all, there's a reaction from the scientific community saying, well, why have you studied Parkinson's disease? Because it's actually not very closely dependent on genes so far as we are aware. So Parkinson's disease is actually an odd condition. Brin persisted and, in fact, has given $2 million, apparently on the basis of his finding, to the Michael J. Fox Foundation, Michael J. Fox being a well-known actor suffering from Parkinson's disease. And the Michael J. Fox Foundation actually gave $600,000 to 23andMe to do the screening. Well, it turns out that 23andMe is actually a company that's run by the wife of Sergey Brin, which is likely, when you realize that, to undermine confidence in the business enterprise that surrounds genetic screening at the moment. So investigate with interest, but be aware. Now, one of the things that I've said or implied frequently throughout this lecture is that most of our genes we have in common with people who may look completely different from us. Black people, white people, different racial constructs. And I've tried to emphasize that, but also at the same time acknowledge that there are quite strong racial associations with some genetically based conditions. And one of them is breast cancer, where it's very well known that among Ashkenazi Jews, the frequency of the two most important breast cancer genes, BRCA1 and 2, is very common. So these are some data. You can see that the frequency of those genes among Ashkenazis and actually Hispanics is rather profoundly elevated among the frequency as compared with the frequency in the white population. And if you are a carrier of those genes, you have something like a 50 to 80% lifetime chance of developing breast cancer. And the problem is that ovarian cancer is thrown into the mix as well. And women who carry both of these genes have a strong risk of ovarian cancer too. And Ashkenazi Jews who have confronted these rather stark statistics have written various gripping accounts of how they've come to terms with that. This is the book by, recently published, Blood Matters by Masha Gessen, an Ashkenazi Jew, and the book review in the New York Times was headed Chronicle of a Death Foretold, which is pretty close to the truth. And in it she writes, "My body had turned against me. All I could do now was to declare war on it myself." And of course what she's referring to there is the medical advice that she was given that she needs to seriously consider having her ovaries removed and her breast removed. And the book describes rather grippingly how she comes to terms with those decisions and what she decides to do. It's a fascinating story, and it's clearly a very tough set of decisions that need to be made. Now, because of the, I'm going to conclude now, because of the strong racial association between genes and disease, a group of people who work in the field back in 2007, last year, decided to get together and write some, what they consider to be guiding principles to deal with the question of race and genetics, including health. And I don't altogether like this list of 10 things that they come up with because it mixes science with various other things. But the ones I've outlined in blue are pretty good. I like 2 to 7. And in fact, point 7 on this list is a key point that underpins this lecture. Medicine should really focus on individuals and not races. Well, I think that's still too strong a statement. I think the more balanced statement is that if you're going to think about the relationship between races and diseases, there may be an association, but beware in that investigation. What we really want to know is what the underlying physiological and genetic basis is for that association, and avoid the racial stereotyping. So sickle cell disease is common in black Africans, but it's not really about black Africans. It's really about people who have been exposed to malaria, and that's Europeans as well. And so if you make your diagnosis on that basis, you risk ending up with the wrong treatments for people who are racially classified on the basis of superficial characteristics. As for number 9, oversimplified science feeds popular misconceptions, well, I hope this present account of oversimplified science won't have fed any of your misconceptions. I hope it may have rebalanced some misconceptions. So in conclusion, 2 general points. One is that race is actually generally a poor guide to genetic diseases, and that's because we share most of our genes with most other people around the world. There are genetic differences between races, and they are linked to a few conditions, but not very many conditions. And most genetic diseases are likely to be common and have common solutions across all peoples in the world. So race doesn't distinguish between environmental and genetic causes. It's not likely to identify many genetic causes of disease, although it will identify some, and it could lead to the wrong diagnosis and treatment, as has often already happened. The second point looks to the future. There is a revolution in genetics going on at the moment, as I said right at the beginning, and that revolution is going to persist through this century. It's going to lead to personalized medicine in some form. That's going to begin with things like gene screening, 23andMe. There'll be many other websites like that. There'll be many other services like that. Some of those services are going to be very useful. How rapidly personalized medicine is going to proceed is not clear, but personalized medicine is clearly the way to go. Rather than racial medicine, if you like, personalized medicine, understanding the genetic makeup of individuals is what's going to be important, but that's going to be the work of the 21st century. Thanks for listening.